Hello viewers, welcome to this lecture on plant groups. In this particular lecture, I shall be speaking on fungal classification and reproduction. The major objectives of this lecture will be to know the major characteristic feature of fungi, to know about their structural features, if there is any structural peculiarity, and we also will be knowing about the different fungal groups, that is the most recent classification of fungi, and towards the end we shall be discussing the different reproductive features of fungi. Fungi are non-chlorophyllous organism which is equidistant from both plant and animal, and they have a pretty old fossil history. About 440 million years ago, the mycelial fungi fossil has been discovered, and as you can see in this picture, there are specific mycelial pattern with septation, and not only that, there are propagules which will propagate from the mycelia which indicate unique vegetative reproductive pattern. Now, as I was telling, the major characteristic feature of fungi are they are non-chlorophyllous, which can be unicellular or mycelial or thread-like. They are associated with animals, that is both invertebrate and vertebrate animals. They can be saprophytic, that is they will help in decomposition, like they produce different enzymes like cellulase and hemicellulase and ligninase, and they grow on lignin substrate, and many of these edible mushrooms they actually grow on this lignin or hay. They can be entomopathogen, that is they are capable of growing on insect, they can kill insect, as a result of which they are unique biological control agent for different insect pests. And this common group is known as Entomophorales or insect killers, and they include the cordyceps. As you can see, they have invaded the ant and they have killed it. Beauveria is a very common fungal pathogen which is parasitizing on caterpillar, and they are protecting crop plants. Apart from that, fungi can also act as necrotropic organism like the sclerotinia, which will develop brown lesion and kill the cells on host plant like soybean. The reproduction include vegetative, asexual and sexual. The vegetative reproduction can be by fission or fragmentation. Asexual reproduction can be by different types of spores which can be both motile or non-motile in nature, and sexual reproduction can be by fertilization or syngamy or gametangial copulation or gametangial contact. As I was telling, the nutrition can be both as saprophyte or parasite, and they are basically very important degrading organisms, so they can help in recycling of complex organic substrate. The fungal classification was given by Alexopoulos way back in the 50s. Subsequently, after that, it was revised by Mims and Blackwell in 1996, and basically many fungal groups have been actually replaced by other names. Not only that, some of the fungal members are considered as fungus-like organism, but they are not considered under true fungi. So the status is like this. There are three major groups. One is true fungi, which is Mycota or Eumycota. The second one is fungi-like organism, which is not truly fungus, and they include the Straminipila. And the third one is basically the slime mold, which are more close to protozoa, and they are called the Myxomycota. So the true fungi or Eumycota, they include the Chytridiomycota, Zygomycota, Ascomycota and Basidiomycota. Whereas the Straminipila, that is fungi-like organism, they include the Oomycota, Hypochytridiomycota and Labyrinthulomycota. And the slime molds or Myxomycota, they include the Plasmodiophoromycota, Dictyostelomycota and Acrassiomycota. So these are the major fungal groups, and I shall be discussing one after another these groups. So we start with the Myxomycota because they are more close to protozoa. So they are multinucleated thalloid body, and they grow in unfavorable condition, and they can produce fruiting body or fructification, and the reproductive structure is represented by sporangia. They can produce haploid spores, or sometimes they can produce gametes, and gametes will fuse by syngamy and will produce zygote. And the example is Physarum. But this particular Myxomycota, they are of different types. Structurally, if they are parasitic, they may cause the clubroot of crucifer, that is the Plasmodiophora brassicae. And very often, anti-amoebic compounds can be used to control this particular organism. Sometimes they look like dog vomit, and as it is called dog vomit slime, which is growing on the tree bark, and the name is Fulligo septica. Sometimes they may cause another disease like the powdery scab of potato, and this powdery scab of potato will produce black lesion on the potato tuber, and it includes the Spongospora subterranea. Or sometimes the particular fungal group or fungi-like organism Mycelia will help in carrying different wheat mosaic virus like this Polymyxa graminis. Next we come to fungi-like organism or the Oomycota, which is commonly called water molds, and they include unicellular cells or unicellular organism, cenocytic and filamentous. Cenocytic meaning the filament is there but there is no intermediate septation. They can be saprophytic or parasitic, and they have the basic food reserve as glycogen, which is important, and they do not contain chitin in the cell wall, and that is the reason why they are not true fungi. And very interestingly, they have diploid or eukaryotic nucleus, and they reproduce by zoospore, and this zoospore may have two types of flagella, that is whip-like or whiplash and tinsel or with sub-branches. And the cell wall is composed of cellulose, and they do not have any particular chitin. They may be marine, freshwater or terrestrial, and they include different organisms. Some of them include the Phytophthora infestans, which is the lead blight pathogen, and it is famous because it caused the famous Irish famine in the 1845, where the potato fields were destroyed by this fungi. They also include the Pythium. Pythium is also pathogen because it causes the damping of disease in crop plants. They also include the Albigo candida, which is the white rust of crucifer. They include the Peronospora causing the downy mildew of pea. But these are all crop plants. At the same time, they can invade fishes, and saprolegnia can actually cause infection of fish, and they can kill the fish by fin rot. Another group which belongs to Oomycota, but they are different from Oomycota and they have some unique characters, and this group is called Labyrinthulomycetes. They are mostly marine, and they are parasitizing on seaweed or algae, and they also can act as decomposer growing on dead plant and animal. And they have unique filamentous structure which will put the cell together, and this unique network of filaments will actually help us to name it as Labyrinthulomycetes. Inside the cell, there is a unique network-like structure which will hold the different membranous structure, and this is called botrosome or sagenogen, and this is a unique organelle present in Labyrinthulomycetes. We come to another group under Oomycota, and that is called Hypochytridiomycetes. They are different from Labyrinthulo because they may be freshwater and marine both, and they have zoospore which with only one anterior flagella. The thallus is holocarpic or eucarpic, meaning the entire plant body will act as a reproductive structure, or part of the plant body will produce the reproductive structure. They can be monocentric or polycentric, and the vegetative structure is having some unique rhizoidal hyphae which actually will help the plant body to survive by getting inside the substratum. Now, let us consider the true fungi, and we start with the Chytridiomycota. Chytrids, it is a small group of fungi having approximately 900 species, and they can grow on aquatic and terrestrial habitat, and they also reproduce by zoospore, and they may be pathogenic. Sometimes this Syncytium endobioticum, which is causing wart disease of potato, or sometimes they may transport plant viruses like Olpidium, and this particular saprotrope, they are growing on cellulose, chitin, keratin. so they can produce all these enzymes, and these enzymes can degrade keratin. What does it mean? It means keratin which is present in human skin can be degraded by these fungi, so they can be human pathogenic as well. Now, there are different types of thalli present in chytrids, which include the epibiotic meaning naked outside monocentric one, which will give rise to zoospore. It can be endobiotic, meaning they are growing inside the plant filament and they develop rhizoidal branches for attachment, and they are mostly growing inside the plant body. It can be holocarpic, that means entire plant body is acting as the reproductive structure, or it can be eucarpic, meaning it will grow in mycelia and part of that mycelia will be representing the reproductive structure. As I was telling, it causes the what disease of potato, that is the Syncytium endobioticum, and this type of growth present in potato tuber is called hyperplastic growth. And this is interesting, the same fungi can also cause fish infection or destroy fishes. They can also cause infection to frog or amphibia, and the name frog chytrid is given to Batrachochytrium because they destroy frog. A group of fungi which was placed under Zygomycota, now it has got a separate division, and that is called Glomeromycota. So Glomeromycota is different from Zygomycota because they produce a unique group of fungi developing as arbuscular mycorrhiza, and this particular arbuscular mycorrhiza will be developing mycorrhizal association with the plant body, and they survive as endomycorrhiza, and there may be 80% vascular plant which will be invaded by this fungi, and they develop this association, meaning the plant will give the sugar to this particular fungi, and in exchange the fungi will supply nutrients and water to the plant root. This particular fungi will have sinocytic hyphae, they will be reproducing with a unique type of multinucleated blastospores, and they produce mycorrhiza. So when we see a particular plant, it is very interesting that when the mycorrhiza develop, you can see the network of root also increases, and thereby the nutrients can be absorbed by this particular plant with the aid of this fungus. And when this VAM fungi, as the name signifies, vesicular arbuscular mycorrhiza, is growing inside the plant root, they really are not parasite. They will grow inside and they produce two types of structure. One is vesicle, another one is arbuscle, meaning network, and by this they will provide nourishment to the plant. Now we come to the next group, which is Zygomycota, and Zygomycota actually is important because it is a true fungi and it reproduces by a unique diploid spore called zygospore. Apart from that, they will also produce asexual spore called sporangiospore, and there may be around 800 species, and they have unique mycelia which is aseptate, and the mycelia is called cynocytic, and they are very commonly growing on fruit and bread, and they are called bread molds or fruit molds. As I was telling, the reproduction is carried out, the asexual reproduction is carried out by dark colored dumbbell-shaped structures called sporangium, and this sporangium actually will have typical classification. Like in Rhizopus, they may be arising from the same point, multiple sporangia from the same point, but in Mucor they are a single one, and the central part, which is columella, is giving rise to spores. Or sometimes in Thamnidium there is an apical one with the lateral branches producing smaller sporangia. Or sometimes in Choanophora they will produce a particular sporangia which is flagellated, and this sporangia will be attached to the central columella in discrete fashion. Now we come to the next group, which is Ascomycota. The division is so called because they produce a unique type of meiospores called ascospore, which are borne inside cylindrical structure called ascus. But this particular group, they may have unicellular yeast-like organism, or they may also have simple mycelial or filamentous structure. But what is unique is they do not have any flagellated cell, and they may have more amount of chitin and less amount of cellulose. Because they are saprophytic, they are capable of producing secondary metabolites in the form of different enzymes and vitamins. but definitely they also can do a unique property, that is the fermentation process, and yeast is famous for this particular activity. The structure, as I was telling, is represented by filamentous mycelia, and this mycelia will have septation, but the septation is having a simple pore which is sometimes guarded by a vesicular structure, and this is called Voronin body. So the Voronin body actually is preventing the leakage of cytoplasm in between the septation. And this particular group of fungi is divided into three major groups. The first one is the fungi which produces the ascocarp or fruiting body, and this group is called Pizizomycotina, and they may include the Naked-Asci and the Clistothecium, meaning the covering is done by mycelial structure which will not give any opportunity for the spores to disappear. Then there may be the Perithecium, which is little bit flask-shaped structure, but Apothecium is typically cup-shaped or saucer-shaped structure. Clistothecium is found in Penicillium, Perithecium is found in Claviceps, and Apothecium is found in Ascobolus. And this particular fungi is growing on cow dung and is also called coprophilous fungi. The second major group or subphyla within Ascomycota is Saccharomycotina, and they are also called the Hemiascomycetes. The Saccharomycotina, they are the true yeast or the Candida. Yeast is a fermenting organism reproducing by budding, and Candida is also reproducing by budding, but it may be pathogenic to human like the Candida albicans. The third group, which is the Taphrainomycotina, previously it was called Archiascomycetes, is including the hyphal fungi like Neolecta, Taphraina, etc. And also interestingly, it has the fission yeast. But very interesting, they have colorful fruit body or colorful thallus structure. The last major group under true fungi is Basidiomycota. Basidiomycota incidentally represent the second largest group after Ascomycota, and they include nearly 30,000 different species, and this particular phylum is called so because they produce basidia and basidiospores. Unlike Ascus, the basidia is actually a club-shaped structure, and the basidiospores are borne outside the basidia in finger-like projection called sterigmata. And this Basidiomycota is having septate mycelia. Not only that, they are having a unique property of clamp connection by which two mycelia can join each other and produce a compact rhizomorph. The septation is complex type because they have a unique guarded septum or guarded pore in the septum, and this type of septum is called dolichopore septum. This particular group is divided into three major subphyla, which is Ustilaginomycotina, Puccineomycotina, and Agaricomycotina. The Ustilaginomycotina is actually representing a pathogenic group of fungi commonly known as smut fungi. And this smut fungi is actually invading the corn, and they develop into intracellular mycelia, they develop into N+N stage. That is, they have three different stages. One is haploid N, one is dikaryon or N+N, another one is diploid, that is 2N. So this karyogamy will continue, then the promycelium will develop and it will germinate, but of course after germination they can reproduce by budding. So this smut fungi represent the first group, which is Ustilagomydis. The second one is the famous rust fungi, and it is called Puccineomycotina, and this rust fungi is important because it produces the dreadful disease of black stem rust of wheat. And there are different spore forms growing in two different host plant. One is wheat, where the Euridia or Tilia and the Spermatia or the basic basidiospores are produced inside the wheat plant, but then they get into, that is, the basidiospores will attack the second host, which is barberry plant, and they produce two types of structures like the pycnium and the acium on the leaves, and thereby this aciospores again will invade the host plant, the primary host, that is wheat. So it is a very interesting phenomenon, and it is called heteroecious rust. It means what? That it will grow in two plants, and when the wheat plant is not there in the field, they can survive during unfavorable condition in the barberry plant. But the third group is mainly the Agaricomycotina, which is called the mushroom. It is so called because they have a unique stipe and a particular stalk, which is called the stipe, and the pileus, which is umbrella-like. The toadstools, the agaricus, the lentinula, different types of mushrooms, and some of them are edible in nature and they are high protein diet. Fungal reproduction mainly can be classified into anamorph, that is asexual stage, and the teleomorph, that is sexual stage. Asexual reproduction, because it is undergoing mitosis, the spores which are generated are called mitospores, and it includes the non-motile spores like conidia, sporangiospore, zoospores are motile spores. And this particular structure, that is the mitospores, will include the unique thin-walled spores or conidia, which are cut off in acropetal or basipetal succession found in Penicillium and Aspergillus. Pycnidia is also present in higher fungi like in Basidiomycota, and it means a sac-like structure where they will produce the pycniospores which will come out in aggregate. But the third structure, which is called the acervulus, this acervulus will actually be growing as a mat on which the finger-like conidiophores are produced, and they produce the conidiospores, or sometimes it produces the other type of spores which is asexual in nature. We come to the next type, which is the sporangiospores, and actually the sporangiospores grow in the form of an apical swelling, which gradually become a club-shaped structure having a central columella and a peripheral sporangiospores, and these sporangiospores will be liberated out when the particular sporangia ruptures by absorbing moisture. The zoospores represented the true motile spores, and this motility is brought about by unique type of flagella which include a single whip-like flagella or branched tinsel flagella, or sometimes there may be two flagella, both are whip-like, or sometimes there may be one whiplash, one tinsel flagella, as seen in Phytophthora belonging to Umycota. This is a unique type of spore which is thick-walled intercalary in position, and it is called chlamydospore. It means when the situation is unfavorable, they will survive as a resting spore, and then with the advent of favorable condition, they germinate to produce mycelia. Sclerotia is also a type of structure which is growing during unfavorable condition, and it is a thickened structure which will help the organism to survive during unfavorable condition, seen in Sclerotinia. But another type of spore is the meiospore. Why it is so called? It is because that the diploid structure which is produced by fusion of two gametes will undergo reduction division or meiosis and produce haploid spores, and that's why the name meiospores come. And it includes the ascospores found in Ascomycota or basidiospores found in Basidiomycota. But in mucor, we have a unique type of reproduction which is called gametangial copulation. It means two mycelia will come close to each other, they develop a lateral protuberance which will fuse and produce a diploid structure or zygospore which will germinate to produce a mycelia again. So this type of reproduction is called conjugation, also called gametangial copulation, because there is no specific difference between the male and the female reproductive structure. This is a unique type of reproduction seen in some ascomycetous fungi like in Penicillium, where there is the male antheridium and the female oogonium, or sometimes the female ascogonium and the male antheridium. What happens is the antheridium will come close to the female, and the female will actually bind with the antheridium with the help of an umbrella-like structure called trichogyne, and then there will be fertilization. Or sometimes there will be direct penetration of the antheridia releasing the male spores inside the female, and the reproduction will occur and it will produce the diploid spore. But this type of structure or this type of reproduction is called gametangial contact. And finally, in higher fungi, as I was telling, that two mycelia will come close to each other and they will fuse. So with the highest possible reproductive group, that is the Basidiomycota, there is simple mycelial fusion, and after this fusion, the nuclei will either fuse or will remain in a paired condition which is dikaryon. and that is the reason why in higher Ascomycetes or in Basidiomycetes fungi, that is Ascomycota and Basidiomycota fungi, there will be haploid N, diploid 2N, and there is an intermediate N plus N or dikaryon. So if the nuclei are compatible, there will be fusion, otherwise they separate out and they grow separately. So in this particular lecture, what I have done is, I have discussed the different characters of fungi, I have discussed the different classification groups of fungi, and finally I have concluded with the different reproductive structures or reproductive pattern of fungi. Thank you.